All right, so experiment 31 is isolation of Lactobacillus. Okay, so lactic acid bacteria. So Lactobacillus is a type of lactic acid bacteria. All right, and what these bacteria do, so yogurt, all the stuff you eat, it's made through the fermentation of milk by lactic acid bacteria. So yogurt just has lactic acid in it, and the way that you make lactic acid is through the fermentation of sugars. Now, many of these lactic acid are gram-positive, and they are aerotolerant anaerobes. Yeah, so, sorry, I get kind of blanky every now and then. Yeah, so they're aerotolerant anaerobes, so they grow in the presence of oxygen but don't require it. And there's so just generally, there's two different types of fermentation. There's homofermentative and there's heterofermentative. So homofermentative just means that you make one product from fermentation. So if you're homofermentative for lactic acid, that means you only make lactic acid. If you're heterofermentative, that means you can make various products from fermentation. And these bacteria make lactic acid as well as other things. And then most of these bacteria are catalase negative. So lactic acid kind of grow kind of interestingly. So lactobacillus can only grow in pH below 6, so kind of in an acidic environment. So in milk, what happens naturally is that in milk, streptococcus then establish themselves first. All right, and they'll ferment sugars up until the pH reaches 6. Right, so they'll ferment sugars until it goes from 7 to 6. And then after that, then lactobacillus will start to grow, and then they'll grow and ferment sugars until the pH gets to about 5, and then they'll die out. So manufacturers, so people who make yogurts and stuff like this, they deal with these special lactobacillus that you can just put in, and then they'll ferment sugars, and then they'll make your yogurt that you want. But it's kind of interesting that it's called a succession, where you have to add something first to get the certain environmental conditions, and then once those environmental conditions are met, then the second thing can start growing, and then this thing can continue on for a while, that you can have several different bacteria grow in succession of one another. So what we did in this lab was that we took a loop of bacteria from yogurt, and we grew it on what's MRS agar, which is selective for Lactobacillus. MRS stands for demand, is the demand rugosa sharp broth. And it's just selective for Lactobacillus. We then took that and streaked it. We took that broth, and then kind of like what we did with the nitrogen fixation, but we took that broth and then we streaked that on an MRS plate, right? And so then you can further select it. So anytime it's kind of a similar thing in that you're taking microorganisms from the dirt, so you mix it with a broth, then take the microorganisms from that broth and put it on a plate. Same thing in that you're taking it from yogurt, taking it from the broth, and putting it on a plate. It's just a way of kind of purifying your culture of the things that you want to specifically select for, while getting rid of all the yogurt particles or dirt particles or whatever it may be. But then we gram stained these bacteria and did a catalase test to kind of indicate if they're Lactobacillus. So it's relatively straightforward experiment with regard to isolation. We're kind of at our isolation stage right here. Last lab we did isolation of Streptococcus. This lab we're doing isolation of Lactobacillus. Next lab I think we're doing Streptococcus. So we're kind of isolating bacteria right now, which is an important thing to do in microbiology.